Not yet.
Yeah.
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 Now it's clear.
Yeah.
So I was talking about the clinical query. So what clinical query you are going to answer with your essay? Then what are the type of sample you are going to have, and how you are going to prepare your sample for that particular experiment or essay, and how you are going to deal with your sample, and how you are going to collect the data which will answer your clinical query, and is there anything further which you can do with that same sample, or you can answer about the prognostication and other things. There is some problem. My slide is not moving now.
Just double click on the screen, sir.
Just click
Okay.
in the center.
Click in the
Okay.
center.
Okay.
Yeah.
So now coming, so as I told you, the first point is like, what is the technology or you are using? So what is flow cytometry? So flow cytometry is a word which, if you divide this word, it will give you the three parts like flow, cyto, and metry. So flow means in a stream, it's like flowing. And then cyto means cell and metry means measurement. So by this technology, what you are doing, you are measuring the physical, chemical, biological properties of an individual cell or a particle, which is in the stream. It is going one by one. So then, so this is the flow cytometry. In this, what you do, that a flowing fluid having certain particles or cells and going through a laser one by one, and you are analyzing various properties which include physical, chemical, as well as biological properties. So why we use flow cytometry in hematology? As I'm a hematopathologist, so I'll talk about hematopathology. So it has a unique ability. So you can analyze thousands of events per second. Then it gives you a power that you can take out a population, particular population which you are going to analyze or study from a heterogeneous mixture. So simultaneously, you can see here, it is a marrow slide. So there are various populations and at the different stages also. So with the help of particular combination of antigens, you can take out the proper population. Suppose you are going to study the erythroid population, you are going to study the myeloid population. So you will just get out your population and you will study the various antigen presentation and the various maturation path of a particular population. And with the flow cytometry help of this, you can comment on the strength of antigen expression, which is very important because so many cells can be positive for a particular antigen. I'll give you few examples when I'll be dealing with the interpretation. So how a flow cytometer looks? So flow cytometer, these are the common flow cytometers which are there in diagnostic, most of the diagnostic labs. So BD and Beckman, they are the most leaders in India. So Pex Canto, this is 8 to 10 color. Then Nevio's Cytoplex, so it is 13 color. And BD Lyrics, it is 10 to 12 colors. Configurations are there. Then what is inside the flow cytometer? So if you see that flow cytometer, inside the flow cytometer, the main parts are fluidic system, then the optical system, which is again, made up of emission optics and collection optics. Then there is an electronic system which will convert your signal, and the informatic system where your signal is converted into the data by analog to digital conversion. And the finally a file format, which is usually we label it as like FCS file or LMD file, they will come out and you will analyze with help of different software, and you can do the multiple analysis like you can histogram, dot plot, contour plot, t-SNE, SPAD, or cluster analysis you can do. So coming to the first part, so what is hydrodynamic focusing? If you say, so hydrodynamic focusing, what you are doing, you are arranging in a file, single cell file, a randomly arranged population. Like if you think about your Mumbai railway station, local Mumbai, the population are randomly going there. But simultaneously, if you think about the population which are going for check-in at the airport, they are going one by one, and the officer, security officer is there, and they are calling you one by one and checking you and let you go in. The same way, this arrangement is there. So if you see this video, small video, the cells are randomly coming, and with the help of hydrodynamic focusing, they are going one by one through a laser. So what is going on? Basically, the sample fluid is injected in the stream of seed fluid. And this pressure difference is making randomly arranged cell in a one by one in a linear fashion. This is very important because you have to analyze a single cell and its fluorescence So cell should come, or at the interrogation point where laser light hits the cell, so cell should come one by one. Again, what are the scatters? So whenever any cell interrupts the laser light, so it will scatter light in every direction. Some of the light is directed in the forward direction. In this video, you can see that forward light which is directed. So any spherical particle or material, when it crosses through the light, the surface of this sphere will divert some of the light in the forward direction. And according to me theory, you can roughly just estimate the size of this particle. So if you see here, cells are coming one by one across the laser light, and the light is diverted, and this diverted light is collected by a channel. And this channel will further create a voltage pulse, and then you will analyze, you will have some data about the forward scatter, which will tell you about the approximate size of this cell. So if the cell, you can see here, if the cell is going one by one, you can see the voltage pulse is generated. So if the cell is small, the pulse which is generated is of small height. Then if the cell is higher size, larger cell, then the size is more. And if you see here that whenever the cell is passing, a dot is created here. So if the cell is large, it will go towards the right side. If the cell is small, it will go towards the left side. So the number of events are collected in a particular way, and this type of histogram will be generated. So here you can see the cells on the right hand side, they are large, and the cells which are on the left hand side were small. Similarly, for the side scatter, if you see that light is scattered in every direction, but it is easier to collect the light at the 90-degree, where you will get the maximum signal from this scattering light. So it will tell you about the internal complexity, the complexity of nucleus, complexity of the granules, and the other vacuoles or material is there, then the scatter will be high. So again, in the same way, light is collected as the forward scatter, the light is collected, and you can see the population with the different scatters. So here, if you see that forward scatter will tell you about the size, and side scatter will tell you about the inner complexity, about the granules, nucleus, and the other vacuoles and cytoplasmic material. So if you will combine, yeah, pardon, sir.
Avinash, can you use the laser pointer also?
Yeah, sir. Yeah. So if you see here, this low side scatter, forward low side scatter, and low forward scatter cells, they are your lymphocyte. If you consider this is a like peripheral blood flow cytometer, and if you see this scatter plot, and the population which is of moderate size and the side scatter higher than the lymphocyte is your monocyte, and the population which is of the same size but the highest side scatter in this sample is your neutrophils. So this is how you are able to segregate the different population in your peripheral blood, and you can label this population as lymphocyte, monocyte, and granulocyte. And this is the power of your flow cytometer. So till now, what we have learned, so the basic principle of flow cytometer is like to gather the light from the various scatters and channels. So we are going to read about the physical properties of the cells by forward and side scatter. Then by the fluorescence, we will detect the fluorescence from various antigens which are on the surface, and we will label them with the fluorescently tagged antibodies, and we will check the fluorescence in different detectors as we are detecting the forward and side scatter. So what we'll do, so here you can see we discussed this already, that forward scatter will tell you about the size, and right angled scatter or side scatter will tell you about the inner complexity of the granules. But then we know that a cell has multiple antigens, and if you use very particular antibodies which are tagged with the fluorochromes, you can exploit this property to know the immunophenotype of the cells. So what happens when the laser light fall on these fluorochrome tagged antibodies, which are attached to the particular antigens on the cell, and so these fluorochromes will excited by the laser light, and they will emit some light, and this light will be detected. So the excitation light is different from the emission light because there is some loss of energy, and we know that this energy and wavelength, they are inversely proportional. So what is fluorescence? Any chemical compound which can be excited by the light and then it will emit some light. So these compounds, they have a unstable electron in their orbit, and this orbit is getting excited with the particular light, or the light is a source of energy, so they will be excited, their electron goes into a higher orbit, and after some time they will again release, because that's an unstable stage, they will release the energy and will come back to the ground state. In this process they will emit some light. So you can see here that this is an antibody which is attached to some fluorophore. This fluorophore will be excited by particular wavelength of light, and then it will emit a particular wavelength of light. So if it is attached to the cell, then this light will be there. So what are the application of flow cytometry? There are numerous applications. So in the field of science and research, ion influx studies, cytokine studies, protein engineering, immunophenotyping, cell viability, microbial assays. So there are numerous applications. As a hematopathologist, I have dealt with immunophenotyping and cell cycle analysis, which we are using for BLLs and other places where we are going to detect the ploidy analysis. So if you see, this is the plot given in a flow cytometry man, we call them Paul Robinson's latest paper, that how you can use and what are the advantages or fruits you are getting from flow cytometry data. So if you are analyzing cells one by one, you are getting a lot of things. You can do cell immunophenotyping, cell cycle analysis, you can detect the cellular functions. So they are all of research use. So coming to flow cytometry in hematology, so if we roughly divide use of flow cytometry in hematology, we can divide it for benign disorders and for malignant disorder. If we call it as for benign disorder, you can again further divide it as for RBC disorders, WBC disorders, and platelet disorders. So in RBC, you can see you can analyze RBC for PNH, for spherocytosis, and for some people are detecting fetal hemoglobin also. Then for WBC, CD4, 8 count, primary immune disease analysis, and for platelet aggregation studies, platelet function studies, storage disorders, people are used. Then coming to the malignant disorders, so they are grossly divided into diagnostic one and the monitoring one. So what you call it as MRD or measurable residual disease. So in hematopathology or hematology, the diagnosis is not from the single thing. It is like an integrated diagnosis. So it won't come from a single modality. It is a integrated diagnosis. So you should have a proper knowledge of your clinical history and clinics, what is there, what are the clinical query, what is the clinical suspicion of. Then what you are seeing under the microscope, it is really very important, and based on these two, clinical history and microscopy, you will choose your panel. Panel is something that you are going to put for your flow cytometry sample. It is a combination of various antibodies which you are going to apply for a particular assay to detect. Suppose you are suspecting acute leukemia based on the morphology and clinical history, then you will put a panel which is going to detect the blast and further characterization of blast. Then you need genetic or molecular analysis, as we all know that recent WHOs, they all are having classification based on genetic analysis only. So in hematopathology, the diagnosis will be the integrated one. So flow cytometry in hematopoietic neoplasm, what it is going to tell us. So what are the indications? So first of all, we'll use for diagnosis and classification, like whether it is what is the lineage, whether you can further classify this disease. So whether it is a B or T or AML, then you will search for the therapeutic targets. So we all are very much aware about the CD20 as a target. We have a drug rituximab. Recently for AML treatment or BPDCN treatment, people are targeting CD123 also. CD33 is also a target. And we are using CAR-T therapies like CD19 targeting or BCMA targeting therapies. Then we are detecting all these hematopoietic neoplasm. So whether we are able to detect some disease at a 10 raised to power minus 3 or 4 level, which is a very good thing by flow cytometry. So if you are able to detect all these things, so for residual disease detection, then finally assessment of biological parameters which are associated with prognosis. So we all have learned about the CD38 expression, JEP70 expression, CD49D expression, and other markers which will tell you about the prognosis of the hematopoietic neoplasms. So after all this thing, then there are few interpretative considerations. So whenever you are going to analyze flow data, you should be very much aware that what are the abnormalities and why I'm talking about this. That if you remember that in your pediatrics or whatever we have learned that there are particular maturation thing. If an infant grows at a particular stage or particular month, he will lose some responses, he will gain some responses. Same happens with our blast population. So when they matures, they will gain particular some antigens and they will lose particular antigens. So there are various maturation curves or maturation path for these things, and that we detect. So basically why it happens, because all these things, all the phenotype is determined by, so if you remember from your 12th biology, what is the phenotype? Phenotype is genotype and external environmental factors. So if your genotype is deranged, then all these antigen expression will be deranged. So what are the things you are going to analyze in your sample is gain of antigen. So what does it mean by gain of antigen? If a cell population, if you are looking for a myeloid antigens and you are getting a bright 7 in this population or bright 56 in myeloid population, that means this antigen should not be there. It is a gain. So this is abnormal for this population. Similarly, abnormally increased or decreased level of expression. So particularly if you see that at a particular stage, CD33, 13, 38 should not be there or should be there with a low intensity, but it is coming as a brighter one. Or at a, like if in a B cell precursor, if they are losing 38, that's a significant thing and you will consider that as abnormal. And then asynchronous antigen presentation. So we all know that a particular antigen should not be there in that particular lineage. So suppose we are dealing with 34 positive myeloid blasts. These blasts should not be positive for 11B or bright CD15, or if we are talking about B lymphoid cells, they should usually not positive with CD34 or 20 should not come simultaneously. So that means it is something abnormal. So we should try to find out the abnormality in the immunophenotype rather than just positivity or negativity. Then abnormal homogeneous expression. So there are various patterns that at some stage that antigen expression should be heterogeneous or variable. But if we are getting that expression as a homogeneous cluster or loss of that antigen or negativity, that means there is a abnormal presentation. So immunophenotypic identification of hematopoietic neoplasm, what it needs a thorough knowledge of normal pattern. Then only you are going to comment on that this thing should not be there at this stage. Then only you will say that this is abnormal because it is not present or it is abnormally present at this blast population or this particular population. Then you can say this antigenic abnormality will tell you about that whether the population is normal or abnormal. So till that, here we have that we learned about the basics, that what are the interpretative, what is flow cytometry, how it works, what is fluorescence, what are the cell, and what we should consider before interpreting any hematopoietic neoplasm or sample. So what happens when a sample reaches to your lab? So first of all, you should think about or you should search about the clinical history and what is the clinical query. So a clinician must have sent you that a sample with a clinical suspicion, whether this is acute leukemia, some chronic disorder, or they are thinking for something else, like in HIV, they are just 1 to 34 or CD4 enumeration or something else. Then what you see under the microscope, if there is a suspicion of hematolymphoid neoplasm, suppose clinician might raise a suspicion of chronic neoplasm, but if you are seeing blast, because it will decide your panel, what panel you are going to put. It's like that histopathology, that what are the antibodies you are going to put that particular further things. So you have to choose your antibody panel accordingly, what you are seeing under the microscope and what is the clinical query. Then you will choose your panel, and simultaneously, if it is the already treated one or this is the diagnostic one, this will help you to put your panel and go for the further analysis. So sample preparation, once you receive the sample and you decide your panel, then what thing you have to consider in your mind, that what are the sample type, what is the cell count, what are the staining method you are going to choose, and about points about the data acquisition, because you are going to prepare the sample and then you are going to acquire the sample on your flow cytometer. So you have various types of sample, but most commonly what sample you are going to have is bone marrow aspirate or peripheral blood. At some center, people are doing CSF fluid analysis, pleural fluid analysis, particularly in TLL cases or other malignancies, then lymph node aspirate for lymphomas, and some people are doing biopsy also. So what is the needed thing is like single cell suspension you need. So how you are going to prepare your single cell suspension from all these things, it is the point. Then you will take the cell count. How it is going to help you, that because people are having different panels. So lab where I work, we have different panels, and based on that morphology and clinical suspicion, we put different panels. So we have acute leukemia panel, we had CLPD, myeloma, BMRD, TMRD, and AMLMRD. And based on that, we have 7 tubes in AL. We are requiring 4 tubes for CLPD, 1 unstained to detect fluorescence, and simultaneously 1 tube for BMRD, 1 tube for TMRD. And accordingly, we acquire the events. So this sample volume calculation is needed, that how much sample you need and how much sample you are going to take further for your sample preparation or your cell preparation. So for that, this formula is there. You can copy it. So sample volume is equal to what is the sample volume required is the number of event you require in each tube, then the WBC count of the sample, and it is multiplied by the number of tubes you are going to acquire for that particular sample. So after that, you have to choose your staining method. So there are various staining methods. Most people are using one of these 3 methods. So whether it is Vulclize stain method, mostly we are using this method for MRD. So what we are doing, we are simultaneously, after taking cell count, we will lyse the sample because we don't need RBC. Our target population is WBC. So we lyse the RBC with ammonium chloride based solution. And then we are going to prepare our cell, centrifuge the solution, wash it, and then we'll stain it further with the cocktail of antibodies. Then lyse stain no wash, we are mainly using for 34 analysis, basically because we need a particular count. So we don't want to lose anything. So we are not going to wash this sample. Then a stain lyse wash method, many people are using this for diagnostic panels. So basically what you are doing, you are collecting sample, and based on that sample, you are preparing a single cell suspension. Some people are using a blocking step if they are having problem of your non-specific binding. So there are many Fc receptor which can bind to the cell surface of monocytes or monocytoid cells, and they can create a problem of non-specific binding. But usually this is not that major one if your procedure is properly done and well standardized procedure. Then what you will do first, that we are going to deal with, suppose we are dealing with acute leukemia. So what we'll do first the surface marker staining. So we will put surface markers, then we will wash it. Then we have to go for cytoplasmic stain also, whereas we know that cytoplasmic detection is necessary, particularly for AML. You are going to do MPO and cytoplasmic CD3 for characterization of whether it is T or AML or B. So what you do, you will fix the cell and permeabilize. You will create holes in the cell membrane, and then you will stain the cell with that particular antibody, so that within these holes, these antibodies will go inside and take the further antigen. And then again you will wash, and then you will analyze. You will acquire this sample on your flow cytometer, and it will give you data, and you will analyze it further. So till now, what we have done, like we have done with cell count, cell type, and cell count, then we stained the cell, then we are coming to the data acquisition. So if you see in this plot, on the left-hand side, there are some tabs and other things. So here, every instruction you can give. So what is here, that this has everything to what to do. So in particular samples in MRE, what is the number of event you want to acquire per second? What should be the speed of your acquisition, whether you want to keep it slow, particularly it is, if you are acquiring for fluid analysis, you will go slow. Then if you are going for acute leukemia or MRD cases, for MRD you are going fast, and for diagnostic cases you are going as a medium speed. So these things you will, so this is your panel when you are starting your acquisition. These things will be there on the screen. But before that, you should check your daily QC, and your in that QC, every range or every CV should be less than 5%. So here everything is green. That means QC is passed, and then you are going to acquire your sample, and further you are going to analyze that sample. Then what are the types of flow cytometer? Oh, what are the types of plot you are going to generate? So histograms, where you will have data on one axis and count on another axis. Then the dot plots, each and every dot represents an event which is equivalent to a single cell. So here in dot plot, it is a bivariate plot. Both the side you are having some of your antigen parameter, and you will detect the event accordingly. Similarly, there are few contour plots and density plot. So the denser the area in the center, and simultaneously the contour plots will tell you about the lines which are limited by the density, but here they are lines rather than the density. So before starting analysis, we must check about the compensation. So you have to check your compensation, and why you have to check it? Because you are using the light, and it is really challenging for that any analyzer, any channel to have that only that particular light. So some of the light from the signal is going to another channel also, and which is leading to this spillover. So you have to correct it, and then you will see that whether your data is properly compensated or overcompensated or undercompensated. Here you can see that this population is going upside. So if you are going to analyze, you will think that this population is positive for this expression, but this is a false positive one. Simultaneously, you should have the knowledge about your spread of population. So some of the population, some of the cell population with that particular channel will be like spread. It is, if you see this angle is going like this, and this is not a proper positivity. This is just like a spread. So before commenting on the positivity, you should be aware what is the spread or spill error in your assay. And then you will have an eagle's eye, like your scanner. You should view or at least 4 plot like this before starting or getting the population further. So first plot is time plot. So this plot is considered as a QC plot, and it is a QC for your fluidics, whether your fluidic system is working fine or not. So you will check for a smooth acquisition, whether with this time there is any interruption, any block, any clog is there, then the data will not be smooth. So it will tell you about that your fluidics is working fine, you can go ahead. Then what you will do the doublet exclusion. So what are the doublets? Doublets is basically in flow cytometer, each event correspond to a single particle. But sometimes two cells get stuck or two particles got stuck and they come as a single event. So it is creating the doublet. So why it is necessary to create, to identify the doublet and take out from your analysis. So what happens that if a single cell is coming like this, it will create a single pulse. But a doublet is coming, it will create a pulse which will be having a higher area, but the height is almost the same. So on this plot, you can easily check out that whether your population is having, whether this doublet is there or not. So with this plot of height versus area, because area is going to increase, but height is not. So you can go for the linearity, and you will create a plot and you will segregate your singlet population. And after getting the singlet population, what you will do, the population which is having low forward scatter but having high side scatter is usually the dead cells. So you will remove all these dead cells and debris, and what you will collect, so this you will create this gate. I labeled it as real cells, and then further you are going to plot all this population on the 45 versus side scatter plot. So these 4 plot: time plot, singlet, viability or real cell plot, and then your 45 versus side scatter. You should be well versed with all these plots, because you have to start your analysis from these plots only. Then on the 45 versus side scatter plot, if you see, you can divide various zones. The population which is in blue color, if you see, this is 45 bright, but the side scatter is very low. So as we already discussed in our forward and side scatter plot, that lymphocyte have a lower side scatter, but the lymphocyte will have a 45 brighter. So this population is your lymphocytic population, or this zone, if you say this is a lymphocytic zone. Then the population which is having higher side scatter than the lymphocyte is your monocytic population. And the population which is in green is having the highest side scatter, and this is your granulocytic population. And the population which is having 45 dim is in red color and is having low side scatter is your blast or progenitor region. There can be various populations sitting here. We'll come to all these areas one by one. So if we think like this as a schematic diagram, on the x-axis there is CD45, on the y-axis there is side scatter. So we'll go from the lymphocytic region to further. So in this lymphocytic gate or lymphocytic region, what are the population can be there? What are the possibilities? So if you see about the possibilities, mostly the benign lymphocyte if you run a normal sample. But what are the other population can come? So the population of B cell lymphomas, T cell lymphomas, mainly lymphocytic. The population, mature population will come here, and then TLGL population. Then we'll come to the monocytic population. So what are the population and what are the population you will find here? There will be monocytes. Some of the monoblasts may reach up to here. Hairy cell leukemia. Why hairy cell leukemia cell will have the higher side scatter? Because their cytoplasm will have a lot of flavonoids. So because of this flavonoids, their scatter will increase and their size is a little bit larger than the normal lymphocyte. So they will sit in this monocytic region. So they may be in continuum from lymphocytic to monocytic region. Then mast cell leukemia, we all know that mast cell have a lot of granules. Sometimes, very rarely, plasma cell neoplasm may be CD45 positive, or their subset may be positive, and that subset may fall here. And granulocytes in MDS. So what happens in MDS, myelodysplastic syndrome, that granulocytic population will lose the scatter. They are hypogranular, and if they are hypogranular, then their side scatter will be low. So they will merge with this monocytic population. That's what is criteria for loss of side scatter is one of the criteria for MDS detection by flow cytometry. Then in this region of granulocyte, what you will get? Normally granulocyte, then the hypergranular variant of APML will come here, but you won't see other population. And with the correlating with your morphology, you will come to know that yes, it is APML. Then if you go to this 45 dim zone, I divided this zone into two colors. If you go through this orange one, it is having a very minimal side scatter and 45 dim. So mostly this is a zone for your hematogones. Then your B cell and T cell leukemias may sit here. Very rarely AML population which is not having any granules and small population may come in this region. Then the main population which is like a little bit higher scatter than hematogone population and 45 is dim and connecting with the neutrophilic or monocytic population, this zone is usually contains your myeloid blast, and the population in hypogranular variant it will come like this, or some people say a teardrop pattern. I didn't make it. And then precursor B cell leukemia lymphoma population may fall here because in some of the cases they might lose a little bit of CD45. They can sit here. Again, in this region, you will find your basophil population, your NK cells, and your PDCs. So your plasmacytoid dendritic cell neoplasm may sit here. So if you are well versed with this plot, and you will see this plot again and again with various analysis and various tumors, and you will start recalling, then you will remember that, yeah, this population is sitting here. Then how we interpret the antigenic expressions. So there are certain antigens which are specific to that lineage. So they are mutually exclusive to that lineage. If we talk about the CD19, it is exclusively expressed in the B cell lineage. So it is mutually exclusive for B cell. Similarly, CD7, it is also mutually exclusive for T cell lineage. What are the co-expression when you say that this is co-expression? So if you analyze CD3 and 7 on T cell population, it will be expressing both. So they are co-expressed. And then some of the antigens, they are not expressed on that particular population. So this population is your normal lymphocyte.
Double negative.
And it is double negative. Yeah. So this population is double negative. So this lymphoid population is non-expressor for 34 and 117, and it will work as a quality control, your internal control for your further analysis, because this population should not be positive for your CD34 and 117. I talked about the strength of antigen. That will tell you about what population is. So if you see this plot, in this plot, this CD38, this is side scatter. On the y-axis, this is side scatter. So this 38 versus side scatter plot, you see here, the population is divided into 3 or 4 zones. So the population which is bright for CD38 is your plasma cell. The population which is moderate is your hematogones. You can see here this population, orange color population. Then the population which is your light blue or aqua is your monocyte. They are also positive for 38. A part of lymphocyte, which are activated lymphocytes, are also positive for 38. So there are 3-4 population which are positive for 38. What is telling about you classifying the population is like the strength of antigen. So flow cytometry gives you this privilege that you can classify your population based on the strength of expression, which is difficult by IHC. If you are putting, then it is really difficult to classify that no, this cell is this. This is the privilege given by the flow cytometry, and this green population is totally negative for that. And if you see about the when you are labeling bright, moderate, so you see this is a beautiful maturation pattern of hematogone. So I plotted it 45 versus 20. So if you see that hematogone 1 will go like this, then they will go. So if you see here that this pattern from negative to this side is your variable heterogeneous pattern. So we usually write in our analysis or whenever we are reporting that this antigen is expressed in a variable form, this antigen is expressed in dim, this antigen is bright or moderate. So it will tell you about the population, that whether this population is there or not, whether this antigen expression is normal or not. If this 45 dim population is showing you bright 20, then it may be abnormal. Then again, coming to the further antigenic expression, if you see, as I told you, on the right side plot, you can see 20 variable. And here, if you see, this 20 is model. So we know that 20 variable expression is abnormal, and it is usually like dim 20 or variable 20 is a finding in CLL. So expression strength will tell you about the disease also. So you should be aware about the normal pattern as well as how you are going to interpret your antigen expression. Now coming to the how you are going to identify the blast. So this is a very old slide from my mentors. So identification of blast, what it needs. I showed you 45 versus side scatter plot. So the zone 45 dim zone is considered as your immature or precursor population zone. So these cells usually have low side scatter and 45 dim. Then they will lack the marker of maturity. So maturity image, they will have marker of immaturity. What are the immaturity markers we are studying, like CD34 and TDT? So expression of 34 in this red population. So this is your blast population, and similarly the TDT positive population. And what are the other markers? Sorry. And this population will, this abnormal precursor population will lack the marker of maturity markers won't be there. So to identify your blast population, what you should have: dim 45, low side scatter, marker of immaturity like 34 or TDT, and lack of marker of maturity like CD11B, 15, or CD20. So for that particular lineage, how you are going to identify your lineage? So these are the list of markers which are related with the other lineage specific markers or lineage markers. To call it as myeloid cell, you should, these cells should be 33 positive, 13 positive. So these are the markers for particular lineage, and you have to mark it up. There is no other option. Until unless we remember all these markers, it is difficult to analyze. So till now we have discussed about everything. If you think about that, flow cytometry is just like a game. So we discussed that we will study the normal pattern and then we'll go for the abnormal one. So what we are going to do, we are going to find the differences from the normal. if we know the normal blast, they will look like this, then it is very easy to search for the differences. So if we consider on the left side, there is a normal blast population, on the right side, your sample is there, then it is easy that you are going to search for the differences there. And this is what we are doing in flow cytometry. We are just finding differences from the normal, whether there is a deviation from normal path, where there is overexpression, underexpression, gain of antigen, etc. So I'm just going for a brief, a single case. So this case is like 46-year male, presented to the clinics with fever and bicytopenia with raised TLC. So this, you can see here, like on peripheral ischemia, there are cells looking like blasts. So I go higher, and these are the blasts. And some of these blasts will show me or rods. Yeah. So anybody will comment on the cellularity of this marrow?
So for that age, hypercellular.
Hypercellular. So how you'll say that marrow is hypercellular?
100 minus the age, sir.
100 minus age. Sometimes people use 100 minus age plus minus 20. That can be useful, right? And then on that low power, I'm seeing a lot of blast and other myeloid population, along with a little bit of erythroid population. So there are some normal neutrophils, then blastoid cells. So I put my panel. Which panel I'm going to use now?
Acute.
Myeloid.
Yeah, basically, you cannot say based on just morphology that this is going to be myeloid. Sometimes some of the rare blast population may be there. So either we will put your alert tube, acute leukemia orientation tube, or wherever I was working, we were using acute leukemia panel. So I put my acute leukemia panel where we were using 7 tubes, right? So this was the MPO. So till now we are sure that this is a MPO positive acute leukemia. But since we have seen that even after MPO positivity, there may be a small clone of T cell or B cell may be there. We cannot rule out. So we'll label it as MPO positive acute leukemia, and we will wait for the flow result. So in flow cytometry, what I got, so as I told you that we will take entire data, we will put the time plot, and we'll do the sequential gating. From this time plot, we'll take this time population here, and we will exclude all the doublets, and then we remove all the debris cells, and we'll took just viable cells. Again, we further clear the population, and we make these clean viability or NJ viability gate. And based on that, you can see this population is going like this. Here is your normal lymphocyte on the 45 side scatter plot. These are your granulocytes. And these are the various maturation patterns. So if you see that your blast population is your positive for what? Can somebody tell me? This red population is
CD34.
34 positive.
34 positive. So if you see here that on the x-axis, what is there? Side scatter. On the y-axis is what? 45. So this population is 45
Dim.
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 And side scatter is
Low side scatter.
low side scatter. So low side scatter, dim 45, falling into your 45 progenitus or 45 blastoid region, progenitor region. So now we know that this population is in progenitor region. Simultaneously, this population is showing us CD34 positivity. So now we are sure that this is an immature population, right? Then what we'll see that how these cell matures. So these are the normal patterns. We have some templates based on the normal control. So this is the way how neutrophil matures, then how monocyte matures. So here we are seeing that this 34 positive population is showing us what? 15. 15 usually should not be there or should be very dim. So this is some sort of abnormality. And the blast number or percentage is already increased. So marrow with increased blast, presenting with bicytopenia, increased blast, peripheral blood is having blast, blasts are MPO positive, and then they are positive for 34 and dim for 45 and low side scatter and showing you CD15 also. So this is like going in the direction of abnormality. So these are the abnormalities. Again, if you see here, so this is the way how neutrophil matures. So they are again 45 dim and they are negative for
16.
Yeah, they are negative for 16. So 16 is a marker for mature granulocytes. Again, they are positive for CD117 also. And they are again negative for 11B. And here, if you see, these cells are showing you what? You can follow my pointer.
7 expression.
CD7.
So what we learned in the interpretative consideration, what is this?
Aberrant.
Aberrant. This is a sort of gain of antigen. And CD15 is there, that is like a asynchronous presentation. Okay, so there is asynchronicity, immature cells, and they gain CD7. So that means definitely this population is abnormal, right? And then this population is positive for 15 and 13 also. And if we see this population is negative for cytoplasmic CD3, positive for MPO, negative for 22 and 79, and dimly positive for, so variable sort of DR, moderate to dim, and dim positive for CD64. And there is no CD56 expression. And there is no CD4 and 8 expression. So where we are leading, now we can conclude our case. And there is no 19, no 10, no CD20. So what will be the diagnosis?
Acute myeloid leukemia.
Yeah, now we can say acute myeloid leukemia. But if we see that in this way, if you see the immunophenotype, this can predict us to a particular abnormality. So there is a data regarding that if a 34 positive population, which is 15 positive, 7 positive, can be 34 positive, 7 positive, 15 positive, and showing you a little bit of erythroid maturation in this. In your previous slide, we saw that marrow is not entirely replaced by the blast. So this could be your biallelic SEPA. So this case was positive for biallelic SEPA on the NGS, right? So this is a AML with biallelic SEPA mutation. So here I'm concluding my talk. So now this is the single case. Further, more cases Kunal sir will take. So from my presentation, what I just want to tell you guys that diagnosis of hematolymphoid neoplasm, you cannot go by a single entity. It's a multidisciplinary approach, and you should include everything possible whenever you are making a diagnosis. And the other thing is, don't be in rush. Don't just put a gate on CD45 dim region and will say that this is your blast or this is the. Try to see the patterns, that what are the abnormalities from normal. What are the immunophenotypic abnormalities? Because immunophenotypic abnormality comes from the derangement of some sort of a genetic damage. If any genetic damage is there, then only your phenotype will change. Then always correlate with other modalities. And if you are thinking that you are missing something or you are not able to connect with the other part, then there is no issue. You can always go back and review your findings. Okay. And the last thing, that when you are spending more time in your flow, you will be happier. Thank you.
Thank you, sir.
Kunal sir.
Hi, Avinash.
So
Thank you for a wonderful
now
talk.
I request Kunal sir to.
Thank you. So students, you have any questions for Avinash or me before I begin my gating session? If you have any questions regarding the talk or anything, please you can ask now. If there are any specific questions, moderators which have come on the group or will come.
The chat box for Dr. Avinash. Do we need to run all tubes for each case after a lot tube, AML, B cell tube, and T cell tube for each case?
No, it is not necessary. If you are running a lot tube and you are working in a resource limited setting, you can just directly go for the positivity of your marker in alert tube, because in alert tube most of the people are having B or T markers, and if you are excluding that this is not a mixed phenotypic leukemia, it is a straight one lineage leukemia, you can go for that particular leukemia.
Just to add to that, what you call a A-lot tube may be different from what I call a A-lot tube. That is where the challenge lies. So while Avinash said if your A-lot tube is extensive and you are using all markers, so for example, the Euroflow A-lot tube is a very extended and a good tube. If you use that marker and then if you have no aberrancies over there, you stick to a B tube for a BL, you are fine. But if you make your own A-lot tube of only 4 markers and a limited B cell panel, limited myeloid panel, you might miss some biophenotypics or problematic cases. So one has to be very careful in which markers you use in your panel. So these questions are good, but you need to define. Sometimes you copy the concept, say for example from Euroflow, without copying the principles or why they are used, which antibody in which panel, which is also not accurate. And like we always taught that for diagnosis of leukemia or a lymphoma, the patient is going to spend money in lakhs. So that few thousand rupees for extra antibodies is something which is, we need to be careful about. So limited panels, I understand resource limited settings are there, but it should not be too limited. Avinash, there are some more questions. What is the phenotype of normal blast in bone marrow? I'll discuss the phenotype of normal blast in the talk ahead. We're not going to the detail of that, but we'll discuss some idea about the same. And Avinash, can you please repeat the specific flow findings in SEPA-AM? Avinash, maybe you can repeat that.
So for that SEPA, these blasts will be positive for 34, 15, they will express 7, and sometimes they will show you 56 also. And on the morphology, you will have erythroid population also, because they say that during this mutation, the cell progeny start diverting towards the erythroid and megakaryocytic lineage. So you will find erythroid population along with blast. Then your phenotype will be 34 positive, 15 positive, 17 positive, and 56 baby positive. So this is a rough idea. It's not like always this phenotype will be positive for SEPA, but you can suggest SEPA studies.
Avinash, can you stop your presentation? I'll share my screen.
Yes, sir. Yes, sir.
So there's a question on how many minimum events are appearing depending upon an acute leukemia or MRD evaluation, and whether to call it positive or negative, that's something subjective, and you need to know knowledge of normals, which I'll discuss just now. So what I'm going to do is,
I just want
go ahead. Yes,
sir, I just want to add one thing.
Sure.
Yeah, so I think that minimum event should be 1 lakh, because I have seen few cases where we are missing some of the blast minute population, which is less than 1%. And when we acquire more event around 1 lakh, we got that minute clone. So otherwise we were missing that clone. So if we are acquiring more event, if we can.
Yes. So I think I agree that, sometimes people go only by the number. What I would like to add is when I say 50,000 to 1 lakh, it should be FSCSC viable events. Yeah. Or in a blood, it should be 45 positive viable events. Otherwise, you'll end up acquiring 90,000 RBCs, unlicensed RBCs, and only 10,000 WBCs. So you'll end up with the problem what Avinash is saying. So these are challenges which you will learn along when you do your analysis. So is my screen visible, Avinash?
Yes, sir. Yes.
Okay, so what I am going to do over the next 45 minutes is actually take you through, wearing the hat of Sherlock Holmes and identifying different populations. And I want your help, the students over there. I'm going to ask some questions. Avinash has beautifully conceptualized a whole journey of a flow sample. My focus is only going to be on the analysis. Okay, he has shown you examples of some leukemia, lymphoma broadly, and shown you some cases of gating and everything. But actually, how do we see in a software? What are these gates? How do they look? What is the feel? Okay, that is what we are going to do. Okay, so my first analysis which I am doing is, like Avinash said, you take the sample type here is peripheral blood. I'm informing you. The second thing I'm telling is what the test we are doing. We are doing a lymphocyte subset test. Okay, so this is the reagent of antibodies which we have put. We have put CD3, 1666 NK cell marker, 45 is a gating marker, 4 is a T cell marker, 19 is a B cell marker, 8 is another T cell marker. So this is called as a 6-color multi-parametric TBNK or lymphocyte subset panel. Now, anyone from the students, what do I mean by this color? 6 color, what does it mean? What is 6 color, 8 color, 4 color, 12 color? Can you tell me? Any one of you?
The number of channels that are there for us to analyze simultaneously.
Great. So in this, your answer is correct. In this machine, like I have shown, I have 12 channels. So my machine is a 12 color instrument where I can put 12 different fluorochromes which emit different colors. Currently, I am using only 6 color in this test. Okay. Anybody can tell me why is this called multi-parametric? Is there any difference between color and parameter? These are the parameters. Okay, it's here in every machine. Forward scatter, side scatter, and the colors are your parameters. So in any machine, if it is a 6 color machine, it will be 8 parameters because you have 2 parameters of forward scatter and side scatter also there. So when I use a 12 color machine, I'll have 14 parameters. So you see FSC, SSC, and then the other 12 colors. Okay. It is very important to know the color I put in the diagram over here. Okay, for example, this is the brown color on these cells, has no connection to the color of the fluorochrome or to the multicolor experiment. This color is like drawing on a piece of paper. You can choose the color you want. Okay, so that is some basics which you should know. So what I'm going to start with is maybe the plots which Avinash told you also the order. The first we start with the time plot. Like he said, this is a QC plot. Okay. We are seeing data acquired over a period of time is stable or not. If there are air bubbles, there'll be a break in this data. There'll be up and down of the data. So the data is stable. Second, he told you that we want to acquire only singlets. We want to acquire cells which are in a 1-to-1 ratio with the area and height. So all these cells which you can see my mouse over here are doublets. So what we do is we draw a gate. To save time, I have drawn these gates and kept. Okay. Don't worry about which software you use. You draw the gate and you give it a color. The next, so this has been given a black color as of now. I have not changed the color. Okay. If I want, I can say choose any color. For example, I'll choose gray. Okay, just to highlight it between the time events and the other events, I can choose any color I want. The next population, typically in any analysis you want to see, is forward and side scatter. Any of your students can tell me what is a forward scatter? Tell you about generally?
Size of the cell.
Size or volume of the cell to be more appropriate. And side scatter?
Internal complexity.
Complexity, the granularity of the cells.
Excellent. So what is a population which will be in this region, which is lower than the forward scatter and side scatter of lymphocytes? What cell population will be there?
So the erythroids or the debris.
Debris. Excellent. And because Avinash showed you, we do a stain lyse wash or a bulk lyse wash or a lyse wash stain, we lyse the RBCs. So if I am going to study for WBCs, like I am doing in this assay, I am studying the basic lymphocyte subset assay. I want to study only the WBCs. So what I do is I put a FSC SSC based viability gate where anything which is below the lymphocyte in forward scatter and which has high scatter, like in this area, this all is debris. Now either I can color the debris or I can color the cells which are my viable cells. So I can even draw a gate on my debris and give it a color if I want. So all these cells which are drawn in now purple color is essentially debris. Okay, I can highlight the events, but I don't want to study these events. What I want to study is only my viable events. Okay, so forward scatter, we are seeing. Any one of you can tell me which is this population in the high forward scatter, relatively slightly higher side scatter above lymphocytes?
Monocytes.
Excellent. This population is?
Neutrophils.
So
Granulocytes.
Avinash, you have taught very well. Okay, so now I am going to go on to my 45 versus side scatter plot. The power of CD45 is it's one single antibody. You can differentiate 4 to 5 different populations without using any other antibody. So if I tell you peripheral blood, by definition, are all WBCs 45 positive or negative? Anyone?
Positive.
Are these my WBCs which I have colored in gray?
No.
No. This is likely to be what cells?
They are 45 dim
Negative cells. So what cell in blood is 45 negative? Any cell which is 45 negative in the blood. platelets and
RBCs.
RBCs. So your unlyzed RBCs, platelet and debris will be all 45 negative. So on any axis, for example, on this x-axis, on the right hand side is positive, on the left hand side is negative. On the y-axis, on the bottom, जी sir.
Can you make a laser pointer?
Okay sir, yes sir.
Or you want to make a pen, or you want to make a pen, whatever.
Sir, how does the pointer come over here?
Just click on the right side of your mouse. Does it work?
Okay. Sir, because that is in PowerPoint.
Okay, ठीक है.
Okay, I am in that software. Sir, is there something on the WebEx platform?
Vivek sir, there is an annotation, something.
Let it be.
Okay, sir, sorry.
Okay.
So this color, if I give gray color to WBCs now, what I have done is ahead of this value, anything which is positive WBCs, I have given a gray color. Okay. Now we are studying lymphocyte subset. So as Avinash was telling you, the property of lymphocytes is that they have the lowest side scatter. So I want to give my color, blue color to lymphocytes. I've already pre-prepared a gate and kept. Okay. I color this population blue and this population name I give as lymphocyte. Okay. As I told you, you can choose a color of your choice. Okay. I can change the color. I can add short name or I can add full name. It doesn't matter. Okay. If you want a different shade of blue which you like, I can use a different shade of blue. It doesn't matter. Okay. So I have gated the lymphocyte. By gating means I have colored it differently and I am going to tell my machine that I only want to study lymphocytes now. Okay. Can you see here? It is written lymphocyte means I can choose in my population. Do I want to study all the WBCs? Do I want to study only the lymphocytes? If I choose only the lymphocyte gate, all the blue color cells are showing. If I choose the 45 gate, all the cells are showing. Okay. So this way I can study which population I want to evaluate at one point of time. Now I know I have put reagents which are T cell markers, B cell markers and NK cell markers. Okay. So I am keeping this box over here. So what we normally do is we gate the T lymphocytes, any color. Okay, I have labeled this just to save time. What are the different subsets of T lymphocytes? Anyone? Amongst T lymphocytes, can you tell me? What would you like to study? Two types of
T regulatory cells,
T regulatory cells.
TH cells, cytotoxic.
Okay. So which is the marker of T helper cells? CD4 or CD8?
CD4.
Okay. So if I want you to tell me how much percentage of CD4 cells is there, what can I do? So first thing I have done here is I have not displayed lymphocytes. See, if I display lymphocyte, the blue color and the pink color cells are being displayed. I have told it I want to display only the pink color T cells. Then I can make a gate around here and say these are my lymphocytes. Not to confuse the two blue colors, I'll give it a different color, say example green color. Okay, I can make a gate around my 8 positive lymphocytes and say give it some other color. Okay, I'll give it say a dark green color, light green and a dark green. So these light green are my 4 positive lymphocytes. They are 45.65%. These are 44%, almost equal in this case. Okay, there are also the leftover cells which are 4A dual negative. Okay, they normally exist. Okay, so immediately you can do this, or the other easier way when you are doing 4A subsets is the software allows you to put a quadrant. I can put a quadrant. Okay, I can choose and tell the software has various features to give me. I can say this is T lymphocytes. I can label the quadrants. So upper left is going to be CD4 positive. Okay, lower right is going to be CD8 positive. Lower left is going to be double negative T cells. Okay, and upper right maybe a few double positive T cells, which I am not showing right now. So if I draw a quadrant, you get the percentages written over there, and you can give CD4 to CD8 ratio. So I have already studied T cell and the subset of T cell, but you say I would like to study the remaining subsets of lymphocyte. Okay, so let's color the non T cells as one color. Okay, we have tried to put a gate. I'm going to reduce the gate of this CD4, CD8, and now come to this gate of 4 versus 19. Okay, let's give the lymphocytes, B lymphocytes, a dark blue color. Now these are my B lymphocytes. So the T are in pink, the B are in blue, navy blue. What will be the remaining lymphocytes be? NK cells. And NK cells will express which antibody?
1656.
So we have 1656. Can you see the NK cells? You can, right? Orange color. Can you see a population which I am now marked in green? What are those cells in green? So these green cells are not CD8 cells. They are 4 positive but not CD3 positive. They are negative for 19. They are practically negative for 1656. Some amount of 16 they are showing. So now you will say, why in lymphocytes non-T cells are expressing CD4? So I have the opportunity, what is called as backgating. Now when we do backgating, whatever color you have displayed, I can go into my lymphocyte gate and see where is the green population line. I am purposely moving the gate up now. Okay. Initially when we started, we did not have these 3 colors. For example, I'm going to remove this T gate. I'm going to remove this B gate. You had the original light blue color over here, right? You did not have the 3 colors. After I put the T gate, you got your pink color. You could back gate and see where the T lymphocytes lie on 45. You could back gate and see where the B and the NK lymphocytes lie. NK, if you compare, have a slightly higher side scatter than the B lymphocytes because they have granules, cytotoxic T cells. So they have slightly higher scatter and little brighter 45. Okay. The T cells in pink have brighter 45 than normal B lymphocytes. Why is it important to know? Invariably, T ALLs are bright 45, whereas B lymphocytes may lose. B ALLs are dim 45 because even normal B lymphocytes have lower 45 compared to T. Okay, so if you know normals, then you can study abnormals. Now let's come to the green color cells. Anybody can tell me what these green color cells are?
Sir, now
Monocyte.
we can ask individually. Otherwise, everybody is not involved.
Okay. Avinash, you can ask one of the students. I don't know. I don't see them on the screen. So if you can ask them. Anyone.
So
What is the
Sabira,
green color population?
Sir, monocyte, sir.
Excellent.
why it is monocyte?
Uh,
You are
What
right,
is?
but why?
sir, it has low side scatter and CD45
So the location on the
dim.
FSC-SSC plot, lower side scatter as compared to neutrophil and above the lymphocyte. That is what you're trying to say, right?
Yes, sir.
So I am telling you this is normal sample. So what did you learn about monocytes? They normally express CD4. So 4
Yes, sir.
is
CD14
also,
and yeah.
yeah, everybody knows 14 is a monocytic marker. And everyone thinks 4 is only a marker of T cells. But CD4 is normally expressed in almost all monocytes. Okay. Now,
Okay.
even in monocytes, this is going a little bit in detail. Can you see there are 2 populations of monocytes? One to the left over here. Okay. Can you see these brighter monocytes which have brighter 45? A significant population of these express CD16 and are actually called as non-classical monocytes. We will not go there, but my idea to tell you is monocytes express 4. So what we do in our lab is, because somebody does not know where to draw the gate for lymphocyte. Okay, it is sometimes very difficult if the lymphocytosis is there are T LGS, they will go up. So you cannot cut. So after removing T cells, we remove the monocytes. Then we get the B cell and the NK cell. So with 6 color antibody assay, you were able to give a report of lymphocyte subset. So for example, if this patient had a 5000 WBC count, of which 20% were lymphocytes, what will be the absolute lymphocyte count?
10,000, 1,000.
WBC count 5000, lymphocyte
To
20%.
11,000.
What will be the absolute lymphocyte count?
1,000.
1000. Excellent.
Louder. Yeah.
Okay,
1,000.
1000. So if now, if my T lymphocytes over here are 56% out of 1000 cells, my absolute T lymphocyte count will be 560. So this way, in our report, we write percentage and we write absolute lymphocyte count. Okay. Any questions on lymphocyte gating, TBNK cells, 45 gating of lymphocytes? Any questions you have? Any of your students? Okay, because just few markers we have seen. Now I'll identify one more population. I'm going to give it a color. Give it some different color. Say orange. And we can give these cells a green color. Okay. Now I can make a plot. I can choose which fluorochrome I want to study. Okay, I want to study, for example, brilliant violet 421 and brilliant violet 510. Can you see I have put any antibody in that? I had shown you my panel. Okay, I'm going to study only WBCs. In my panel, in this sample, if you see, there is no antibody in the 421 channel and no antibody in the 510 channel. Okay, can you see a WBC population over here? Is it positive for both or negative? Can you see this population?
Yes, sir.
Okay. Do you think it is positive for 421 and 510 or it is negative?
Positive or not?
Positive, sir.
Looks positive, right? And it is on the diagonal, telling me that equal positivity for 421 and 510. But we have put no antibody. So this is autofluorescence. And one of the easiest way to identify this population is autofluorescence. Which population will that be? Look at the FACSC plot. Okay. Where are these cells lying the orange color? I am putting my pointer there. They have a scatter higher than neutrophils, lower than neutrophils.
Higher than you.
Higher than neutrophils, right? Okay, I want to change the axis. Give me a second, sorry. Can you see this population better now? Orange color. It is higher than neutrophils. Similar level of 45. What can this population be? Which has higher scatter. Higher scatter means what?
More granular cytoplasm.
Which cell in the blood has more granules and high scatter? More than neutrophils. Thick color granules.
Basophils.
Eosinophils.
Eosinophils. Okay. So these all are eosinophils which are highly autofluorescent. So the violet channel or the blue color channels where there is autofluorescence is maximum, I use that channel to identify my eosinophils. So we identified eosinophils, you identify green color neutrophils, which are high scatter monocytes you saw. Basophils end up lying here. For basophils, you will need one more marker, 123, to ideally identify the basophils. Retrospectively, you can. But in this assay, we have not put those antibodies to identify my basophils. Can you see an empty region over here?
Yeah.
Okay. Do you see any cells over here? Practically nothing, right? I am drawing
No.
a gate. Practically, there is nothing over here. Okay. Dr. Avinash told you the name for this region. Some cells which lie here normally. So what is that name? Progenitor region, right? Normal progenitor. So in peripheral blood, you will not normally see progenitor. In the marrow, you can see normal progenitor. What abnormal cells fall here? In a case of leukemia, what cells are they?
Blasts.
Blast. So some people call this as a blast hole,
Blast.
HOLE. Okay, because they say if you have a large population in this region, which is below the neutrophils to the left of the monocyte and the lymphocyte, this is the progenitor or the blast hole. It's of extremely important significance to us hematopathologists that you always analyze your progenitors. And that is why that photograph or the slide which Avinash was saying, where he was showing different populations which lie normal and abnormal in the progenitor region, is important for us to do. Okay. So in a given sample, we could identify 5 or 6 different subsets with a few number of antibodies and give ourselves an analysis. Any questions or should I move to the other case? Avinash, anything you want to add?
Sir, in the one of the plot, no, CD19 versus CD3,
Yes.
initial plot, sir. Yeah.
Yes, here it is.
So anybody can comment about that this 19 positivity is there or something else is there?
In the blue population, Avinash?
No, sir, pink population.
Okay.
CD3 positivity.
So I'll show you, give you a clue now. So there is what Dr. Avinash is trying to tell you. I'm sorry, this got stuck. Okay. If you compare in the T cells, there seem to be two clusters. There is the 8 positive cluster and there's a 4 positive, which is 8 negative. When you see it together with CD3, it appears that these cells have slightly dimmer 19 more than these cells. That's because Dr. Avinash has explained the concept of spread and overlap. Okay, with experienced eye, we can see that, okay, these are overlapping colors or close fluorochromes. So we are seeing some amount of spread around. Avinash, this is what you are saying, right?
Yes, sir. So I just want to tell them that whenever you are going to report, you just go through your essay, see it, where it is coming, what it is, because we know that 19 is a exclusively B cell marker. And why CD3 positive T cells will show you 19. So we have to go back and check whether is there any compensation error, whether there is any spreading error, or otherwise somebody will say that these T cells are having dim 19 or subset of T cell is having 19. It is not like that. Now you can see it is a properly, a spreading error.
Y shape.
Yeah.
So there are ways to do correction of compensation. We will not head there for today's talk. But the idea was to show that these spreads happen. You should carefully interpret. So it's a very valid point, Avinash. Thank you.
Sir, I forgot to tell one thing.
Yes.
Can you please elaborate about that axis?
Yes.
Linear logarithmic. So because you changed one.
Okay. I'll discuss on that point. It's a very good point.
Yeah.
Okay. So in life, if you want to measure elephants and ants on the same scale, can you measure them on the same ruler? If you want to measure their width or height, you cannot, right? But if you want to measure different types of ant and maybe a cockroach, you will say, okay, this ant and cockroach you can measure on the same scale. Same way, when we measure populations, we need two type of scales. One is called the linear scale. So in this, the forward scatter is on linear scale. What does the linear scale mean? Linear scale means the difference between this 50 to 100, 100 to 150, 150 to 200 is equal. Okay, if this is 50,000, because this is 50 into 1000 over here, you may not be able to see that. This is channel number 50,000. This is channel number 60,000, 70,000, 80,000, 90,000, 100,000. Every time there is a bit scale over here, the gap, there is an equal distance. However, in a logarithmic scale, when you move from 10:2 to 10:3, it means it is moving from 100 to 1000 to 10,000 to 1 lakh. So the distance between the same units is different. It is 1 log. Here you can measure very small to negative signal like an ant to very large signal like an elephant in the same scale. So in spite of them being very far apart in distance, that is 10,000 to maybe the value of 1, you can still measure them in the same scale. So whenever the intensity of the signal is so wide apart, so bright a signal and so negative signal, you need a log scale. Whereas in scatter, they are not very wide apart. The cell channel may range from 50,000 to at the most 200,000. Okay, the multiple is only 1 to 4 times. You can simply do with a linear scale. So there are different scales available on your software, linear and logarithmic. You also have a bi-exponential scale. Like this actually ahead of 10:2 is a log scale. Below 10:2, can you see the 0 and minus 10:2? This becomes a linear scale. So when both scales are expressed or seen on the same line together, they also call it as a bi-exponential scale. And the softwares allow us, for example, in CD19, I can make simply as a log scale. Now you see that 0 and the 10⁻² plus 2 disappeared. Now this is all log scale. Okay. Then I can go to linear scale. You cannot separate the data. because negative, positive, everything has come together. You don't know where they are lying. You cannot separate them or visualize them. Now to separate them or visualize them, I need to convert my CD19 into a log scale. Still some data appears to be on the negative, touching the baseline. So I go to the bi-exponential scale. Now I can separate very nicely negative from positive. So these are the ways of using different type of linear log and bi-exponential scales in flow cytometry. Avinash, should I go to the next case?
Yes, sir. Thank you, sir.
Yes, thank you. So we are going to randomly go to some cases and see what we have in this case. So this case, it came to the lab, it had some abnormal mononuclear cells. Like Avinash had asked you, is it leukemia or lymphoma? What panel will you give? It's a very important question. But sometimes it is very difficult to tell what panel to give because you cannot make out whether this is leukemia, whether this is lymphoma. It is very challenging. Okay, so in a child, if there are very large blasts which go with the leukemia panel, sometimes you have to use both the panels. So that is one point. Anyways, in this case, we had a lot of blasts and we ran a panel. But I wanted to focus on normals first. Okay, can Avinash, you can tell one of the students to answer. What is this blue color population?
Yeah, Bimbit.
This is 45 positive, side scatter plot. I have two populations, one blue and one pink. I want one student to tell me what is the blue population, one to tell me what is the pink population. This is a
Bimbit,
peripheral blood sample.
Sir, the blue population is the lymphocyte, sir.
Excellent. And the pink population?
This is the granulocyte, sir.
Excellent. Okay, so when I am visualizing my B lymphocyte, is there anything in the blast hole? In this empty region, can you see a large population here?
Yes, sir.
Let's color it in red. Okay. While I am showing you red, I want you to focus only on the blue and the pink first. Okay. My red is a blast. Correct? Now, can somebody tell me, can you see the blue positive normal mature B lymphocytes? Yes. Can you see them? Can you see the negative lymphocytes? There are lymphocytes which are negative for
19.
19. Excellent. Okay. Now, I want you to focus on one cell population. Wait. Change the marker for you. Okay. So right now we are seeing some B lymphocytes in 19. You're also seeing the same lymphocytes in 20. Correct? And the B lymphocytes are 10 negative. Normal B lymphocytes are positive for 19 and 20, negative for CD10. Correct?
Yes, sir.
So now you have internal positive control for B lymphocytes. Okay. If now I focus on the blast, can you tell me what is the markers for the blast?
Vinay.
Two plots. Red color population.
Vinay. Dr. Parul.
So this red population, do you call it positive for 10 or negative for 10? Let's make it simple.
Positive for 10.
Excellent. For 19, positive or negative?
Positive for 19.
Okay. Now, even if I remove this quadrant, you will call it positive. If I call normal lymphocyte as moderate, will you call this bright, moderate, or dim?
Bright.
Bright.
Normal population of 19, I call it moderate. Okay. So if I were to draw a straight line, the intensity is similar to normal. Correct? It is not crossing the normal, so I will still call it moderate. Okay. Now, normal neutrophils is pink, is 10 positive, dim 10, there is moderate, so this becomes bright. But for lymphocytes, this is still, for 19 positive, it is still moderate. Now, can somebody tell me, is it 20 positive or negative? This is 20 positive lymphocytes, these are 20 negative lymphocytes, these are 20 negative neutrophils.
20
Now,
negative.
20 negative.
20 negative.
okay. So now if I tell you're right, majority population 20 negative, but if I use my internal control, Okay. I told this is bright, this is negative. Can you see this population which is a little bit
bright.
heterogeneous dim expression of 20? And I'm going to give it a green color for you to visualize. Can you see?
Yes, sir.
So this is dim heterogeneous expression of CD20. Okay. Now I'm going to ask you one more similar. So the next student is 34 positive or negative? In majority blast, you have to tell me, is 34 positive or negative? Sorry, I moved the blast population. Sorry, here it is. 1 second. I'll move it to the center of your screen. 10 versus 34 plot. Is 10 bright? What about 34?
Negative.
Negative.
Majority of the blast are 10 negative, which is excellent. Again, a very small population here may be expressing dim 34. Okay, so these are kind of things which you start noticing, especially when we want to look. But as Avinash was saying, is it spread? Is it non-specific binding? Is it a channel which is the problematic? Is there debris? Is there autofluorescence? We have to be careful of all such things when we interpret. So you have told me 3 or 4 markers over here. It is 10 positive, 19 positive, 34 dim in a subset, 20 dim in a subset. Okay, let's see 38. 38 is nice, moderately expressed. What is the diagnosis? These are blasts which are 45 dim to negative, which are expressing D markers 19, a bright 10, and they are 34 practically negative, 20 practically negative. Any diagnosis? You are seeing sheets of blast on the marrow, on the blood, sorry. What would be your diagnosis? Is it a leukemia? More than 85% blast.
Yes.
Yes,
Okay.
it is.
Is it myeloid leukemia?
No.
Is it T leukemia? Is it B leukemia? What do you think it is?
B leukemia, sir.
B leukemia.
B leukemia. Would you like to see any other markers or you don't want to do it? Would you want to see other markers? So do you, if a child comes, do you put only one tube or do you put the other tubes also? You put all tubes, right?
Tubes.
A tube and myeloid tube. So you should see all other markers.
MPU should be done.
One, you could do MPO and other things, but you should at least basically see your T tube markers and your myeloid tube markers.
Yes.
So when I start with my T tube, I can show you these are the reagents put in my T tube. 8, 7, 3, 4, 45, 56. So NK and T markers which are input. Now I am always never interested in seeing the blast first. I am always interested in seeing the normals first. Okay, otherwise how will we learn? So I have gated my normals again. Blue is lymphocyte. And if you see in our lab and in every lab, everyone uses similar colors continuously. So that your eyes get used to that color and pattern. Okay. So if I use purple for neutrophils or pink, I continue to use pink. If I use blue for lymphocytes, then most of my panel will be blue only. Okay. So first things first, are my lymphocytes looking okay? Because I need to check whether my staff has put the antibody in the panel. Okay. It is very easy to say blast positive, blast negative in a very good panel, but how do you know antibody is there or not there? So first what we do is, in this population I said show me the lymphocytes. It showed me the lymphocytes. I color them. I color the T lymphocytes pink. I color the NK cells green because I know normal NK cells are 7 positive, surface C negative. But you, in the previous assay of TBNK, we studied NK cell marker is 1656. Can you see the green color cells positive? Few of them, 7 and 1656 positive. Are you
Yes.
able to visualize them? Okay. So T cells are 7 and 3 positive. NK cells normally are surface C negative, but positive for 7. So now which will be the lymphocytes which are negative for 7 and 3 in this brown color? What are they likely to be? T cells are over, NK cells are over. What are these cells?
B cells.
B cells.
So you didn't put any marker, but you could study that these are the B cells, these are the NK cells, and these are the T lymphocytes. Correct? Now, if I put a gate on the blast, I know my internal controls are working. Okay? And I say, show me the blast also in this sample. Okay? I'll see the blast now. Now you can confidently say my red color population is negative for 7, negative for 3. What is the expression of 56? Practically negative. What is the expression of CD4? Negative. What is the expression of CD8? Let's put CD8 over here. Negative. Now you don't expect a BLL to express T cell markers, right? But you can see good internal controls, know your normal patterns, know that your antibodies are working, and that the blast are T markers negative. Now I have got B markers positive T, but I need to go to my myeloid tube. Again, my first job in the myeloid tube is to study normals. So what is normal? How do I know? So I get my blast, okay, and I start seeing my markers, okay. So anybody is 33 positive in the blast or negative?
Negative.
Negative.
How do you know my staff has put 33 in this blast, in this tube or not? What if I didn't put an antibody? Is 13 negative or positive?
Negative.
Negative.
64
64.
negative.
Negative.
36 practically
Negative.
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 117.
Negative.
So everything is negative. Have you put antibody or not? So you first get lymphocytes. Okay, I get it my lymphocytes. Lymphocytes are also negative for 33 and 13. Normally also, right? So what population can I get to check if 33 or 13 was put? Is there any normal population in this plot which I can get and study if 33, 13 antibody was put or not? What do you think neutrophils they express?
This should be gated, sir. Neutrophils
If
will
I get these
express.
neutrophils, can you see internal control in 33, 13 now?
Yes, sir.
Myeloid cells, neutrophils and myeloid cells are positive for 13, 33, positive for 64. They are not immature, so they are negative for 117, which is normally so. Now you know 33 and 13 are working. Then when you put the blast, you can confidently say, yes, my myeloid markers are absent, HLA-DR is present. Also to note, HLA-DR is present also on normal B lymphocytes. HLA-DR is a marker which is MHC complex 2 marker present on antigen presenting cells, APC. Anyone which are the antigen presenting cells?
Dendritic cells.
Excellent. Monocytes, B lymphocytes. All these cells express HLA-DR. Okay, so normal B lymphocytes express DR. Hence, it is very common for a B ALL to also express HLA-DR. So this patient had B markers 1910, HLA-DR positive, 45 which was deemed to negative. So we labeled this case as B ALL with absence of TN myeloid markers. Any question on this case? Avinash, would you like to add something?
Kunal,
Yes, sir.
two things. One, I request students, sometimes there is a background noise, students, at least the 4 or 5 who are live. So make sure if you have to talk, you can mute yourself. Second, Kunal, did you tell them 20 was negative? Why you still calling it B cell ALL?
Yes, sir. I'll go on to that. Very good point. So even 34 was negative and 20 is negative. Still, we are labeling it as BLL. Why? Because mature lymphocytes express B, and when cells are developing, what we call as progenitors of B cells are also called as. One of the students, can you tell me the name? What is the terminology used for progenitors of B cells?
Hematogones.
Excellent. So hematogones, when they go through various stages of development, stage 0, stage 1, stage 2, stage 3, they develop expression of 20 only in stage 2 and 3. Okay. So if the arrest in the maturation happened in the stage 0, 1 or 2, in the early stage, they may not express 20, but the presence of 19 with a bright CD10 along with other markers like 22, 79 is also a marker of B cell. So this is important what sir asked, because on IHC we do not have 19. Okay, you will do blast which are 34 negative. You will say 20 negative, 34 negative, 3 negative, MPO negative. I don't know what this is. Okay, but if you put markers like TDT, they can be positive in blast, most common in the ALL blast like T and B. You should use CD10 And if 20, which is the maturity marker, is negative, the B marker used in IHC is PAX5. Okay, so this is where knowledge of normal phenotypes in normal and abnormal both helps, and correlation between flow and IHC. Okay, so while 20 is present on normal mature B lymphocytes, in the blast it may be negative. Please keep this in mind, especially on IHC. Okay, whenever in a child or a leukemia, if you get 20 negative, do not rule out BLL unless you have done CD10 and or at least a PAX5. These two markers are very important. And remember, 10 can be present also in, what is the name of 10? Something with C.
ALA.
ALA. What is the full form?
Common ALL and
Common ALL means it's present also on BLLs and
TLL.
TALLs. So alone 10 is not enough. Okay. So 10 with CD3, CD7, TALL. 10 with 19, 79, 8, 22, PAX5, BALL. Okay. And blast, like normal hematogens, move from stage 1 to 2 and 3. As they mature, they lose CD34. They have 10. Then they start acquiring 20. There are different stages of maturation. For want of time, we cannot go there today. But that is what is also knowledge of normal patterns of development in a bone marrow. So blast can be 34 negative also, both in BLL, TLL as well as in AML. Okay, but this location on the CD45, this is very important. Now, if 45 is bright over here, then I'll be worried whether this is a mature lymphoma, mature cell origin lymphoma, or if 45 is dim to negative like this, I am more worried about a blastic malignancy like a leukemia. Any questions on this, students? So I am going to move to one more patient. Okay. Now this was an adult patient who presented with a lymphocyte count of around WBC count of around 20,000. Okay. And I am going to study some populations. And as usual, my first job is to identify time gate, singlets, and I go to forward and side scatter. Can you see the forward scatter and side scatter? Correct? And this blue color is coming from the gate on. What is this population?
Lymphocytes.
Excellent.
Lymphocytes.
Okay. I'm going to color this again. What is this population which I'm coloring now?
Monocytes.
Monocytes.
Monocytes. And the population above that?
Granules.
Neutrophils. Okay.
Neutrophils. Because this is peripheral, this is neutrophils, there will be some eosinophils scattered above. But this patient had a 15,000 WBC count with almost 75-80% lymphocytes. He is a elderly male who is walked into the clinic and he has lymphocytosis. So now morphologically it looked like mature lymphocytes. So we put a CLPD panel. Okay. In our CLPD panel, we want to study B lymphocytes, T lymphocytes. And within the B lymphocytes, we want to study whether these are polyclonal, so we put kappa lambda, or monoclonal. We also study, sir.
What is CLPD panel? What is CLPD?
Yes.
Lymphoproliferation.
Student, what is the CLPD panel? Avinash had covered partly that we use
What is
different panels
CLPD?
for
What is
leukemia and
CLPD? CLPD, what is it?
full form of CLPD.
Chronic lymphoproliferative disorders.
Excellent. So you have panels, morphology based panels if you use. You have acute leukemia panel, a panel for lymphoma or chronic lymphoproliferative disorder, which is typically also directed towards mature B cells and or mature T cells. Then if you have a case of myeloma, you have a myeloma panel. If it's a case of residual disease, you have a minimal residual disease panel. Like I showed you lymphocyte subset, my panel was different. So every disease entity may have separate panels which have to be validated in your lab, and you should know what is normal in them first. Okay, so I'll get these lymphocytes, study them, and I want to study my normal lymphocytes. So what are these lymphocytes in light blue color? What are they expressing? Between 19 and 5, what are they expressing? Are they positive for 19 or negative for 19?
19 and 5.
Positive for
For
19.
both 5 and
What about CD5 expression? The blue color ones? Negative, right? And the green colored ones are T cells or NK cells?
NK.
The green lymphocytes are T cells or NK cells? They are CD5 positive. Anyone?
T cell.
T cells.
T cells. Excellent. So the leftover cells, which are neither B cells nor T cells, will be
NK cells.
NK cells.
likely NK cells. Now, is these three populations covering all the cells, or there's a major population lying outside of it?
Outside.
Outside, right? But let's study normals first. Okay. Normally, B cells, are they all kappa, all lambda? Are they, what should they show? Some kappa, some lambda, right? Can you see the normal blue colored cells? Some of them are kappa, some of them are lambda.
Lambda.
T lymphocytes, negative for kappa lambda. They don't express light chain, right? They are negative. Now, what if I get these 19 and 5 positive lymphocytes? Now I am seeing two populations, and with the knowledge of normals, I'll say, okay, look at this purple pink color population which I have gated in the 5 and 20 plot. This population is my normal polyclonal population. Now what happens to this light blue population? Is this kappa, lambda, both, or only one antibody?
Lambda restriction is there.
Lambda restriction. Compared to normal lymphocytes which express bright lambda, what will you call this intensity? Moderate, dim, or moderate to dim variable? What would you use? This is bright, which is normal.
Moderate.
This will be moderate, so this will be moderate to dim, right? It's sort of heterogeneous. It's not lying as one laddu over here. It's lying like a hot dog which is spread all over. Okay? So it's going from moderate towards the negative region. And the kappa is empty. So this looks like a abnormal population. Why? It is expressing B cell marker 19, co-expressing T cell marker 5. Compared to the purple color cells which are 20 part, which are polyclonal, it is also expressing, showing light chain restriction, means clonality. Now, what is the 20 intensity of these cells? Anybody? Compared to normal, which is bright, I'm going to change the scale a little bit so that you can see it a little better. So the scale is very tightened. Okay. So, this quadrant I can keep here, somebody will call it negative. If I keep here, somebody will call it positive. That is where you need experience. And the quadrant doesn't matter. That's why you need to know normal T lymphocytes are negative for CD. So I'm going to remove the color. Okay, first. I'm going to put only normals. Can you see the normal lymphocytes now? So now if I was to put a quadrant using the knowledge of normals, I would have said, okay, the quadrant looks something like this. Okay. Because my normal T lymphocytes are negative for B, my normal B lymphocytes are negative for 5, then I know this is my quadrant. Now I can see my population. Now compared to the normal B lymphocytes, what about these blue lymphocytes? Are they expressing 20 or they are not expressing 20?
Dim to moderate.
So I would say dim to negative, or dim heterogeneous, or dim variable. Okay, so you have a bright 45 population. So let's see, where does this population lie over here? Because is this lying in the bright 45 region? Yes, it is. I'll zoom into this plot. So I'm doing backgating now. Okay, I'm going to remove, sorry, I'm going to zoom in this gate. Okay, if I remove this color, you will not see, but now if I put this color, can you see purple is lying here, green is lying here, and this majority population, which is your abnormal population, light blue, is lying in the bright 45 region. Slightly lower than normal T lymphocyte, but still bright 45. So it is not in the dim 45 progenitor region. It is a mature B lymphocyte on morphology, looking like mature B lymphocyte. It is a bright 45. It is co-expressing 19.5. It is showing light chain restriction with dim heterogeneous lambda, and it is expressing dim 20. What do you think we are dealing with, leukemia or lymphoma? First thing, CLPD or acute leukemia?
CLPD.
CLPD, mature B lymphocyte showing light chain restriction. Now, can we subtype the CLPD? Do you need any more markers? Do you know any different subtypes of lymphomas in peripheral blood? Any one of your students? Different types of CLPDs.
Mature B cell lymphomas.
In the mature B cell lymphoma, can you tell me 2-3 different types?
So follicular, CLL,
Excellent.
SLL, DLBCL, mantle, marginal.
Excellent. In which lymphoma is CD5 positive?
Mental.
Mantle.
Mantle cell lymphoma.
SLL.
Any other
5
lymphoma?
positive.
Any other lymphoma which is 19-5 co-positive? Most common lymphoma in peripheral blood. Yeah, louder. Somebody said it louder. Mantle, yes, 5 is positive. Any other lymphoma which is CD5 positive?
So CLL, SLL.
Excellent. CLL, SLL. So how do you differentiate CLL, which is also 5 positive, 19 positive, from mantle cell, which is also 5 positive and
23.
So 23.
23 and cyclin.
23, cyclin D1,
D1.
SOX11. Cyclin D1 works better on IHC. SOX11 works better on IHC. As Avinash was saying now, there is advantage of flow. Not only I can tell it is positive and negative, you also told me the normal 20 is bright and the blue cells is the dim heterogeneous. Mantle cell lymphomas normally express very bright 20 and bright light chain, whereas CLL commonly express dim heterogeneous 20 and a dimmer light chain. That is two of the features. In addition, we need additional marker, like somebody was saying 23. Now there are additional markers like 200, 43. Nowadays we use a lot of 200 and 43. CD200 expression is practically present in almost all CLLs and absent in all mantles practically. So let's study another tube which we have put. In this tube, we have put markers of interest which are CD200, 19, 23, 45, 40, 90 and 45 again. So similar gating we have done, okay, where we have put gating on B cells. Now if you see normal B lymphocytes, okay, again I want to study only normals. Normal B lymphocytes in peripheral blood are normally 43 negative and all lymphocytes T and NK cells are 43 positive normally. Okay. Now normal B lymphocytes, okay, are normally expressing, majority of them show moderate heterogeneous 200. Majority are 200 positive, some are negative. Majority. Okay. Normal lymphocytes may show variable 23 and are normally expressing 49D also. Okay. Then I will get my T lymphocytes. T lymphocytes express 43, majority are 200 negative. I'm going to put 43 marker over here and study them together. Okay. Majority are 200 negative, 43 positive, and majority T lymphocytes also express 49D. Now I come to my cells of interest. Now can you see these blue lymphocytes are positive for 43, go positive for 43 and 200. They express very nice CD23. Okay, and I can put 19 over here to see 1923 co-expression. Can you nicely see that 19 intensity is also slightly lower than that of normal lymphocytes? Okay, and these abnormal B lymphocytes are 43 positive, 49D negative. Okay, so now you have a combination that the B cells were showing 5 positivity, but also had dimmer 20 with presence of 243. So what is the diagnosis they want to make, CLL or mantle? What do you think this is?
TM residents.
CLL.
CLL. Okay. So CLL expresses 243, dimmer 20. Okay. So this case was to show you normal cells or polyclonal, how to read the intensity of an antigen, how to define clonality, how to look at mature B cells, and to separate between mantle and CLL. We used not only the intensity, we also used the presence of 243. If both are present, it is along with dim 20 and the presence of 5, that is likely to be CLL. If 200 was negative, 20 was bright, then you would favor mantle cell. You would confirm it with cyclin D1 by IHC or translocation 1114, which is also looking at cyclin D1 by fluorescent in situ hybridization or FISH. Okay. Also, the importance in flow cytometry, not only we diagnose, so we diagnose the B cell lymphoma, we subtype the B cell lymphoma. We said this is CLL. In CLL, there are some prognostic markers like 49D and 38. If 49D and 38 are absent, it is good prognosis. If 49D and 38 are present, it is poor prognosis. In this sample, compared to the normal B cells in purple and the normal T cells in green, the B cells are 49D negative. So good prognosis as far as 49D. I'll go to the first tube where we had 38, which I didn't highlight to you. Okay. We'll put CD38. Okay. So majority of the cells are 38 negative. No one asked me about CD10. 10 positive B cell lymphomas are which lymphomas?
CLP.
Can you tell me one lymphoma which is 10 positive?
Follicular, DLB.
Excellent. So is this lymphoma 10 positive or 10 negative? What do you think? Is it CD10 or CD19? Negative.
Negative.
How do you know antibody is there or not? What if the staff missed putting the antibody? So I had showed you in the previous case, you can use some normal internal control for CD10. Which is that? What population normally expresses 10?
B cells.
B cells, hematogones express, mature B cells don't. Any other population? Do monocytes express 10? Do neutrophils express 10? So normal granulocytes express CD10. Can you see this? The same CD10 is present even in your normal IHC sample. If you do IHC, CD10 will be present on neutrophils also. So be careful. Just because it is brown color IHC does not mean it is positive. Okay. I have the confidence to label my blue cells as negative, my tumor cells as negative, because first I study my internal normal controls. Mature B lymphocytes are 19 positive and 10 negative. Mature neutrophils in brown are 10 positive and 19 negative. Then when I get my tumor, I can confidently say, yes, my tumor is negative for CD10. So identify both normal negative, normal positive, validate your panel, then show your expression. So in any plot, first identify normal, abnormal is very simple.
Great.
Okay, so this was a 10 negative, 5 positive BCLPD of mature cells expressing DIM20, presence of 243, we labeled this case as CLL. And we said, because this patient has lack of 38 and 4090, this is a good prognosis for the patient. So the role of flow cytometry was diagnosis, typing, subtyping, and prognostication, all in one case. Okay. Sir, do we have time for one more case?
Yes, Kunal. Yes.
Okay, I'll quickly go through this case. We will go through 3 tubes. This is also suspected blast on flow cytometry on the morphology. Okay, there you can identify the red color blast. They were around 50% on morphology, and here also in the 45 dim region, I am seeing around 56% population. We are seeing very few monocytes, nothing in the green, few cells in the myeloid region in the pink color population over here, and you can see some mature B lymphocytes. So one by one, I want the students to tell me the markers present over here in this case in the blast population. Okay, one of you is CD10 and 19, positive or negative? Pick. 20?
Negative.
Negative.
20 negative.
Okay, 38.
Positive.
Some moderate positive, little lower than lymphocytes. 34.
Negative.
Is it homogeneous expression or is it sort of a heterogeneous expression? See, I'm coloring it for you.
Heterogeneous.
So it's sort of a heterogeneous variable expression of blast moving from moderate to negative. Okay. So B markers 10, 19, 20 negative, correct? Let's move to the T tube quickly. Okay. Similar colors and gates. Okay. My focus always lies in the normals first. So first I want to see whether my antibodies have worked or not. So yes, I can see my normal T lymphocytes in purple over here. I can see my normal 4:8 ratios. It seems all my patterns are there. Then I will put a gate on my blast and start studying the blast population. So the blast are negative for 3, negative for 7. Okay, I'm going to change the markers to 56, positive, negative. 56 and 8, are they positive or negative?
Negative.
Negative. And you can see background internal controls, right? Then CD4 is left over here. Negative.
Negative. CD.
You can see internal controls of normal T lymphocytes, 4 positive, 8 positive. So you know confidently that these are negative for T. So B negative, T negative, let's look at myeloid markers.
Myeloid marker.
So anyone, what about CD33?
It's positive.
Positive. CD13?
Positive.
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 CD64 over here?
It diffuses.
13 and 64?
Positive.
Positive.
Positive, right? Okay, let's see CD117, immaturity marker. Positive? What
Heterogeneous.
about CD36?
36.
Practically negative in the blast, right? Majority of the blast here are 36 is a marker of erythroid cells. Okay, also marker in platelets as well as some megakaryocytes and monocytes. Okay, HLA-DR positive or negative?
Actually, negative.
Are you sure DR is
Negative.
negative? Has the antibody work? Can you see some normal cells over here, B lymphocytes, which are DR positive? So you know antibody has worked. So this leukemia was 34 dim to negative heterogeneous with DR negativity. Okay. So now, will you label this as BALL, TLL or AML? What do you think this is?
AML.
AML. In AML, does anybody have a clue if 34 is dim to negative and HLA-DR is negative? Do you suspect any cytogenetic or molecular abnormality? Has anyone heard about 34 and DR negative leukemias?
APML, sir.
APML can have 34 and DR negativity. So it is very important here for us to see morphology and look for which translocation defines APML.
PML RR.
What is the number if you want? Translocation in the number?
1575.
Excellent. Suppose I tell you the morphology was monocytic like, it was 1517 negative, and I did NGS testing and I got one mutation positive. It is not APML. And that mutation positive cases also have DR34 negative very often. Any name of that mutation anyone knows? Starts with the capital letter N. N for Niagara.
NPM.
NPM.
NPM1 mutation. So those mutations are also often DR34 negative. So what idea, like when Avinash was telling you about SEPA, what he was trying to tell you is we are making diagnosis, we are looking at patterns, we are also looking at can they be predictive of good prognosis, poor prognosis? Can they be predictive of abnormal translocations or mutations? Because certain mutation translocations have a well-defined phenotype. So this is how we do gating in practice. Okay, this is how I have shown you a case of AML, FEL, replication, etc. I will shortly go through everything. We showed you some normal patterns on TBNK, but this is the broad concept of gating. So open your eyes to normal. You read a lot about abnormals, try and identify how to gate populations, give them nice colors, and compare the plots to identify abnormals. I think we'll stop there as far as the cases is concerned, and we'll be happy to take any questions. Avinash, sir, if you want to add anything.
Just want to add one thing that in this AML case, try to focus on the side scatter.
Yes.
So if you see the scatter, it is going towards the comparing with the monocytes towards myeloid. So this is the first clue for you that this could be myeloid cell or the cells which are having some granules or other thing. As the 45 is dim and the scatter is high, so this is a clue for you to that this population could be myeloid.
So, Kunal, there are one or two questions in the audience.
Yes, sir.
If both kappa and lambda are either positive or negative, can we still call it a lymphoma?
So, good question. One, kappa lambda both negative. So I'll open this case first to show you that question. Okay. Okay. Now I'll see if I have some normal cell. Yeah, I have. Okay. Let's study only kappa and lambda over here and answer that question. Okay. And I am not interested in studying my tumor. I am interested in studying only my normals. Okay. So this population of lymphocytes, which was neither 19 positive nor 5 positive, which is this gray color population, I'm going to gate that in the kappa lambda. Okay. And then I'm going to gate this monocyte population in the kappa lambda, which is P1. Can you see the green color population is both positive in the diagonal in the center? Okay. If number one, you have to identify that is B cells. If I see a population of B cells which is 19 positive, which is absent kappa lambda, first I have to see that has kappa lambda worked or not. Okay, because sometimes processing artifact, because there's a lot of protein in the serum, you need to wash the sample, there are a lot of other artifacts. I need to know whether kappa lambda has worked or not, otherwise I'll be stuck. If it has worked, like in this case, where normal sample is showing kappa and lambda polyclonal, and my abnormal tumor cells are lying in the negative kappa lambda, you can use that also as a surrogate marker of clonality, but with experience and after acquiring a lot of events. This could be due to antibody epitope variation or the tumor cell not expressing it or the processing variation. Generally, when it is dual positive over here, it's a processing artifact or there is not enough separation. Or if the sample is old, there is stickiness of both kappa and lambda, because anything on the diagonal means 1:1 binding. Now, normal monocytes express kappa and lambda. Sometimes people call it non-specific binding. Actually, it is not non-specific. It is specific binding because monocytes express a receptor called FC receptor. FC receptor is what binds to kappa lambda. And because the FC receptor is common for both kappa and lambda, both bind equally, so you get a 1:1 binding. So even if I don't have normal B lymphocytes, if I have monocytes in the center, I can be very confident kappa lambda has worked. Similarly, activated neutrophils can sometimes also express kappa lambda. Or in this case, this is autofluorescence in FITC, so we'll not get there and complicate matters, but this is how it comes. But normal cells, if they work, then that is great. And with the help of normals, with the help of monocytes, you know that this is lambda positive, this is in the center, this is polyclonal. I hope that answers the question.
So there is one more question that for flow cytometry, which sample is better, bone marrow, peripheral blood?
Flow cytometry can be done from any sample, whether it is a CSF, body fluid, peripheral blood, or bone marrow. Obviously, if the disease is in the marrow, okay, and a sample, for example, in a severe pancytopenia patient, if you have no blast in peripheral blood, it is possible the marrow is having 90% blast. I will say marrow is better. But in a patient of CLL, where there is 50% sample, 50-60% abnormal lymphocytes in the blood itself, I don't need to do a marrow to do flow cytometry. So if you have disease cells in the samples, you will pick it up. Obviously, if you have only 2% blast in the blood and 80 in the marrow, I will prefer the 80% sample if available. If not, you can still do it and subtype it, but lesser the blast, lesser the numbers, you need more experience, wider panels, and more carefully in subtyping these patients.
I think, Kunal, there are no more questions right now.
There's one more question now. How many minimum blasts are required in the sample for getting proper blast cluster?
So this how many minimum blast you are talking about morphology. So in MRD we pick up 0.01 to 0.001% blast. In flow, it all depends upon the number of events and what is the target. If it is a leukemia sample, you could do with 5% blast and give a good answer, and you can fail with 5% blast. It all depends on the experience. In a lab which is doing extensive MRD, even 2%, 5% blast is enough. In a beginner lab or person with lesser experience, you might not have the confidence to label 1 or 2% blast as normal versus abnormal, especially when you use limited panels. So that's where, the wider the panel, more the experience, combination with morphology, flow, biopsy, IHC, cytometry, all together as a comprehensive approach is important. Unfortunately, in our country, one person sees aspirate, second sees flow, technician runs the flow, plots come to the doctor may not go to the machine, biopsy read by a fourth person. That is not the ideal way of reporting hematopathology. Even if it is 3 different people, the 3 people need to talk to each other. Okay, even if you are in one lab, you need to go to all 3 departments for the case and then report it. That's where your learning will also happen. So even if your seniors are not doing it, you students should start doing it. That's my take-home message. How much time the sample for flow should be processed? The faster, the better. Normally, we say anything between 24 to 48 hours is good. For precious samples, we do process up to 72 hours. But if you see degeneration, lack of viability in various ways, one should be careful in how you interpret it, especially at very low percentage of abnormal cells. Minimum cell count in peripheral blood, this cell count varies. Okay, so if I have 80% blast